Hey there, biology enthusiasts! Today, we're diving into the fascinating world of how cells eat, specifically the endocytic pathway. You might already know that cells are amazing at exporting molecules, but did you know they're equally talented at bringing stuff in? Let's break it down. First up, we have endocytosis, the process where cells internalize substances from their environment. It's like the cell's way of gobbling up nutrients, hormones, and other important molecules. There are two main types: bulk phase endocytosis and receptor-mediated endocytosis, or RME. Bulk phase endocytosis, also known as pinocytosis, is essentially the cell taking a big gulp of its surroundings. It's non-specific, meaning it internalizes everything in the extracellular fluid, including solutes. Think of it as the cell's way of recycling membrane components. But the real star of the show is receptor-mediated endocytosis. This process is highly selective. Specific molecules, or ligands, bind to their corresponding receptors on the cell surface. These receptors then cluster in specialized regions called coated pits, which are lined with clathrin, a protein that helps form vesicles. Imagine clathrin as the scaffolding that shapes and supports these vesicles. Once these vesicles form, they pinch off into the cell with the help of a protein called dynamin. Dynamin wraps around the neck of budding vesicles and, through GTP hydrolysis, completes the scission. After internalization, the clathrin coat disassembles, allowing the vesicle to fuse with early endosomes. Endosomes act like sorting stations in the cell. They acidify via V-type H+ ATPases, which helps dissociate ligands from receptors. Early endosomes sort materials to different destinations. Some receptors are recycled back to the plasma membrane, while others are sent to late endosomes and lysosomes for degradation. A classic example of receptor-mediated endocytosis is the uptake of low-density lipoproteins, LDLs, which deliver cholesterol to cells. LDLs bind to receptors on the cell surface and are internalized into clathrin-coated vesicles. After fusion with early endosomes, the receptors are recycled, and LDLs are sent to lysosomes where cholesterol is released for cellular use. Now, let's talk phagocytosis, also known as cell eating. This process involves specialized cells like macrophages engulfing large particles such as pathogens or cell debris. The cell extends membrane projections around the target, forming a phagosome. This phagosome then fuses with a lysosome to create a phagolysosome, where the ingested material is degraded. Interestingly, some pathogens have evolved to evade or exploit phagocytosis. For instance, Mycobacterium tuberculosis can inhibit fusion of phagosomes with lysosomes, allowing it to survive inside the cell. In conclusion, the endocytic pathway is a vital counterpart to the cell's biosynthetic secretory pathway. It allows cells to internalize, process, and respond to their environment efficiently. Endocytosis and phagocytosis are essential for nutrient uptake, signal regulation, and defending against pathogens. These processes not only maintain cellular integrity, but are crucial for overall organismal health. Thanks for watching. If you enjoyed this deep dive into cell biology, don't forget to like, comment, and subscribe for more fascinating science content. Catch you in the next video.